Okay. So this is part 2 of DNA, and this is going to be DNA replication. So not when we're doing transcription and translation yet, just how do I get new strands of DNA. As you can tell, I'm no longer at school. I'm at my parents' house. But there'll be a series of 3 videos. The video I recorded on Friday, this video, and one more that you'll need to watch for Monday. So refresher, when we have DNA, DNA is... Okay, so when we look at DNA, it's my sugar backbones and my phosphate, and then my purine and my pyrimidine pairs, so A, T, G, and C bonded with hydrogen bonds. The important thing to remember here is that they are in opposite directions. If we remember, it has to do with the way the phosphate is angled, but it's like a 3 to 5 direction. Always remember you go to the littlest number to the largest number. That's the direction they're organized in. There are 3 possible hypotheses for how DNA replicated when they first started thinking about how do we get new DNA. The first was conservative. Basically, the parent DNA made a complete copy of itself in the form of a daughter strand, and then after each replication, you always got an identical copy of the full parent DNA strand. The second is semi-conservative model. This went along with the idea that you would split the DNA in half. You'd get a copy of one half and a copy of the other, and that would be your daughter DNA. And then the final was dispersive, and the idea was that the daughters were always a mixture of chunks of the original parent DNA. Messelson and Stahl did a experiment that was very similar to Watson and Crick, and to the folks who looked at macrophages, viruses, injecting them into bacteria. They made one strand of the double helix radioactive, and then they looked at where that radioactive material ended up in the new bacteria when they were dividing, and they found out that it is the semi-conservative model that is actually true. Again, this is the experiment. They basically put some 2 bacterias, mixed them together, and then labeled them with like a fluorescent. They let them replicate. They centrifuged them and let them replicate again. And again, what they saw was that always the semi-conservative model was true, or that was the case, that they found strands of DNA that were half of the original DNA and half new DNA. This was the model they proposed. Basically, they believed that the first step to replication was the separation of 2 strands. Each old strand functioned as a template, and so the nucleotides start plugging in along that open template. Then the nucleotides are formed with hydrogen bonds, and they create the new backbone, and so each DNA helix contains 1 strand of old DNA, 1 strand of new DNA. When we talk about prokaryotic replication, this is very different than eukaryotic replication. What we know about prokaryotes is they have circular DNA, and so replication occurs in both directions. So it starts at one point and it moves in opposite directions, as you can see in the images. That's very different than eukaryotic DNA, which is in the strand form. Again, we look at this. DNA replication does occur simultaneously, but it's going to be in a certain direction. So if we look, basically what happens is there's 3 replication bubbles that are visible in DNA when they looked at a hamster cell. And so what happens is DNA occurs away from the bubble until they meet, and then they have the 2 new DNA strands. Because the DNA is so long, we have to have multiple regions of replication happening at once instead of just happening in one place. When we start thinking about replication, helicase are the enzymes that are responsible for unwinding it. Remember, case or aces are always going to be enzymes. Heli is like the helix. It's responsible for splitting up the DNA. And this little graphic is kind of cool. So since DNA is a double helix, there's this tension that causes it to tangle as it's unwound by uh, by helicase. Think of it as like a spring or something like that. When I unwind wire off of a spool, its natural tendency is to like tangle because it's used to being wound in a certain direction. If you've ever unwound wire off of like a spool of something. So topoisomerase, topoisomerase. Um, 1 and 2 different enzymes. They basically are responsible for holding those two strands apart and keeping them straight so that the new daughter cells can show up and bond. Topoisomerase. Guys, it's like 8 o'clock. It's early. So nucleotides are added on as what we call triphosphates. Then the nucleotides are added, and then the 2 phosphates are chopped off. So if we look, or if we remember, there's only 1 phosphate on the parent strand. It's the nucleotide, the sugar, the phosphate. When they're added, they have multiple phosphates, and then we cleave off 2 of them so that they are then left. Again, we would imagine that that releases energy, similar to ATP losing a phosphate. Something that's important, and now that we're talking about the direction, is that synthesis always occurs from the 5 to 3 direction. So synthesis actually occurs opposite to what we would think, right? 3, 4, 5. It occurs from the 5 to 3 direction, which is why when we think about it, the DNA strands are oriented 5 to 3 and then 3 to 5, so that synthesis can be happening in opposite directions in the same space all the time. So 5 to 3 direction, nucleotides, all you need to remember is that nucleotides are always added to the 3 end. This allows for synthesis of a new strand to occur in one direction on the bottom strand and another direction to happen on the top strand. So we have a helicase, we have an enzyme that keeps the strands separate and untangled, and then we need something called a primer. The primer is DNA polymerase, and it attaches the complementary nucleotide to the 3 end. So DNA polymerase is the actual enzyme that is building the sister strand. So what we need is, at the very beginning, we need like a small chunk of nucleotides that signal that this is the start of the DNA. This is where you start linking stuff. And so the primer is what we call primase, and DNA polymerase is physically building this. So if we look at the image, we see primase is this enzyme that signifies that this is the start. It lays down a little bit of RNA, and then the DNA polymerase can notice that that is where it starts, and they move down. So the RNA nucleotides are laid down by a primase before DNA polymerase begins DNA replication. That's kind of a summary of what I just said. So we have something called the lagging strand. If we look at the top right corner, basically the overall direction of replication is occurring from 3 to 5, right? So, from 5 to 3, right? So we're constantly adding new DNA in a certain direction. For one strand of DNA, that's going to be really seamless, right? Because it's going to just be able to go along as the zipper opens. But when we think about it, the bottom strand, or our lagging strand, because DNA synthesis has to happen in the opposite direction, if it keeps going, it's going to meet the end every time. And so what ends up happening is we have to add what we call Okazaki fragments, which are little fragments of DNA. So it's a lot less seamless in how it makes it. This animation shows the leading strand, which is the strand that allows for very seamless replication, is being synthesized very easily, whereas the lagging strand requires chunks of DNA to be put down. The enzyme ligase ties them both together. So we've talked about all the different enzymes. Another important part of this is our telomeres. Once DNA has been replicated, there's a problem. The usual replication machinery provides no way to complete the 5 ends or to kind of chop them off. What? I'm recording a video for my students. Um, so the usual replication, there's no way to complete the 5 ends after the RNA primer is removed. So repeated rounds of replication produce shorter and shorter DNA molecules. So we think about the RNA primer. If every time I replicated that parent cell, but I didn't replicate where the RNA primer is, each time I'm going to get a shorter DNA strand. And eventually I would start losing important material. To compensate for this, the DNA adds repetitive sequences of DNA at the tail end. They're non-coding, so they don't code for any proteins, but they're called telomeres, so we can maintain the length of the DNA so that we don't end up with a shorter one every time because it doesn't code over that RNA primer. So if we look at the 1, 2, 3, fourth down, there's that gap. We fill in that gap with telomeres. So to maintain this in gametes, the shortening of telomeres would cause really serious problems because if chromosomes continue to get shorter during replication, essential genes would be missing. An enzyme complex called telomerase, which telomerase is the enzyme that creates telomeres, catalyzes it and basically lengthens the DNA. So telomerase makes telomeres. So sometimes there are errors in DNA replication. It occurs every once in about 10,000 base pairs. It's really not bad, but when you have 6 billion bases, it's a lot of mistakes every time the cell divides. And so DNA repair systems can repair about 99% of the mistakes, which is why we don't all end up mutated all the time. Basically what happens is something called the thymine dimer distorts the DNA. It basically pulls it apart. Nuclease enzyme cuts that out, DNA polymerase repairs it, and DNA ligase kind of seals it off. And so that is it for DNA replication.